So we are now recording, and thank you very much, Juliana, for agreeing to give a Build a Cell seminar, and it's all yours.
Hi, everyone. Thank you, Katie, for the opportunity for giving to our group to show a little bit of our work. As you can, we'll be able to see, it's a very multidisciplinary group. So the title of our project is about programming biopolymer and biomineral production for living structural materials. And the idea that we started here is by having this multidisciplinary group where we were able to make living building materials as a novel composite for construction. And it's based on the idea that microorganisms living can stay alive in these biomaterials, producing in the living biomaterial in these bricks, which we call brick. And by these microorganisms being able to produce biomaterials that we are of interest for our project, these allow these bricks, these LBMs, to have increased mechanical properties that we're going to talk a little bit more about. So the basic idea of these little bricks, this novel composite for construction that we are working with, it's based, we use here bacteria to produce the biopolymers and biomaterials that we are of interest. And we let them grow and then add sand for the construction purpose. And to bind everything together, we add hydrogel. In this case, we are using gelatin. And we have these nice little bricks that we can use with different properties. So the idea came from the use of traditional materials in construction, where they usually we use from limestones to cement to concrete. So this comes from the natural environments, and they can be resource and energy intensive to manufacture and also contribute to greenhouse gas emissions. And this is a big problem because, you know, as we all know, our populations are growing and we need to construct more and more. And this can have a toll in our environment. So the goal of this project was to develop a hybrid living biomaterial, or LBM, that would have both biological and structural function. And so we would merge in the traditional construction materials that have property, structure, and process, the biology here, where we would go from the nanoscale with, in our case, the bacteria, to that can produce biomaterials of interest. So we can have construction materials that can be easy scaled up, and also with increased complex structures. And the idea is that in the future, have these bricks, these LBMs, in the field of construction being used. So as I mentioned, this is a highly multidisciplinary project. So I'm going to start talking about it. My name is Juliana Artier, and I'm the microbiologist of this project. But we also, in the spirit of having a multidisciplinary project, we decided that it would be interesting to give you a little bit of a view of how our project is by having two other people presenting. Later, Rollin will be talking about biomaterials, and Bruce will be talking about the mechanical performances that our bricks have. So the idea of our bricks here, like I mentioned, uses biomaterials, in the case here, biominerals production by bacteria, where we know that several different types of bacteria can precipitate calcium carbonates, depending on how their bioenvironment is, depending on the chemical conditions of the environment. But the bacteria is important to induce calcium carbonate precipitation by increasing the microenvironment pH, and also provides nucleation sites for crystal formation, which can be different polymorphs of calcium carbonate from calcite, aragonite to ferrite. And this process is highly dispersed in nature. It involves the indirect activity of different metabolic pathways, as you can see reviewed here, from photosynthesis to different other, even methane oxidation, where the indirect products are responsible for increasing this microenvironment pH. And since we have high concentrations of calcium on that specific environment, calcium carbonates can be precipitated. We were specifically interested in first ureolysis, which is the degradation of urea. This is one of the most well-studied pathways in biomineralization. It's so well studied that if you Google ureolysis, it will give you Micp production. And the advantage of this process is that since we know very well the enzymes clusters that are involved in this process, we can tune and change how these enzymes are being active inside of the bacteria cell, which leads to an indirect change of the environmental pH and precipitation of calcites. It's mostly common and well-known and well-described in heterotrophic organisms, so they need complex carbons to survive. 𝗕𝗶ecie 𝗴𝗿𝗺𝗺 that can produce spores and survive in things extreme environments is a well-studied one. But we also studied some of the changes on engineering 𝗕𝗶ecie 𝗴𝗂𝗰 that received the operon from 𝗕𝗶ecie 𝗴𝗺𝗺, where we saw that we had a good nanomechanical properties by simply changing how much calcites or calcium carbonates can be precipitated by modulating the expression of the enzymes in the organisms. And we saw that by tailoring both the shape, we also tailored some of the nanomechanical properties. And this can be of highly interest for our products, for our living building material. The other process that we also studied is MICP, or calcium carbonate precipitation by photosynthesis. The advantage of this is that it uses autotrophic organisms that don't need complex carbons. They are also studied in the sense of the biogeochemical importance, because they are they have an important relationship to the global carbon cycle from different geological periods. And the way this photosynthesis would be important to change the pH in the environment would be that since the cells capture the energy from sunlight and use that for fixating CO2 here in the carboxysomes, the cells during this process also send a hydroxyl group to the outside to balance the ions here. And with that, it alkalinizes the cell surfaces. Again, with the increased concentration of calcium, the cells are able to indirectly inducing calcium carbonate precipitation. What's also known about the photosynthetic process is that both the EPS being produced by the cells and also their S layer has an important feature on the nucleation sites, and also on crystal shapes depending on the negativity or positivity of the amino acids involved. The challenge with working with photosynthetic organisms is that, since you can see here, there's not a direct enzymatic pathway like there was in the urea degradation, where specific enzymes could be targeted for genetically. So that's a little bit of a challenge in this part of the project. However, it's really well known that cyanobacteria can form different calcium carbonate polymorphs. In our study here, previous people from our project showed that 𝗕𝗂𝗃𝗃 702, which is our model organism, can induce calcium carbonates, and you can see these beautiful crystals with exactly the space where the 𝗕𝗂𝗃𝗃 was embedded first. But it comes with the challenge of how can we tailor these polymorphs, as we did with the Escherichia coli urea degradation pathways, to increase their nanomechanical properties. We can also explore different cyanobacteria strains that we're going to talk a bit later. So one of the things that we're doing in our lab to try to understand how the physiological state of the bacteria is involved with the crystals production, we're using time-lapse microscopy so we can follow real-time formation of the crystals and how it affects the growth of the bacteria. And if we leave these crystals to grow further later, we can see them forming structures like we saw in the SEM first. And by using quantitative MATLAB protocols that we have previously established, we can follow the growth of the bacteria and how it's affecting its physiological state. And we can bind this with Raman analysis that are traditional to understand what kind of polymorphs and are being produced by the bacteria. So we are trying to understand a little bit more about how the photosynthetic process is working in the MICP production. So in our work here in our platform, we were focusing on two different organisms, both 𝗕𝗂𝗃𝗃𝗃𝗃, which is a model organism that we use in our lab, and it's a photosynthetic cyanobacteria, and also the Escherichia coli that has been previously engineered, receiving the 𝗕𝗏 operon for the urea degradation enzymes. And we use this technology of MICP to increase the material mechanical properties of our bricks. And one of the problems is that because these bricks are going to be used later for constructions, they need to dry, and we need to keep the bacteria alive under these extreme conditions. So we know from our previous studies, published studies, that if we have more humidity in the bricks, we have higher amount of cells alive. But if we want better structural integrity and we need to decrease the humidity, we face this challenge of bacterial viability in the living biomaterials, and we want to keep them alive because of the idea of living building materials. So as you can see here in this graph, if we follow the number of viable colonies in the bricks through time, if we keep the cells under low temperature and high humidity, the cells can survive even for up to 30 days. However, to increase the mechanical properties, if you want to dry them more, for example, using the ambient relative humidity, like a lower relative humidity and higher temperature, the cells cannot survive. So that was one of the challenges for the project. So we came up with the idea of adding these small sugars like trehalose, which is a very common additive in pharmaceutical and food industries to increase the stability of the products. It follows the idea of vitrification theory. That's one of the theories involved in the project, where these small amounts of sugars can form kind of this glassy matrix that immobilize the proteins and other nucleic acids and keep the proteins stable. Similar idea to what we know from amber that is very well documented. So what we did here, we dried the cells, just the cells for initial tests to see how we could improve the biological survival under desiccation from this 𝗕𝗂𝗃𝗃𝗃𝗃. And we added increasing concentrations of trehalose, and we saw that once we reached about 200 millimolar in our cells, we were able to highly increase the survival from compared to when we don't have any trehalose being present. And if we compare to another cyanobacteria, Anabina 7120, which is considered a moderate desiccation tolerant, we can see that simply by adding trehalose here, the darker colors, we can increase the survival of the cells even under 30 days after desiccated. And we can also increase the survival under stressful conditions, for example, under high light. For example, Anabina 7120 can only survive under very low light, but by adding this simple sugar, we could increase their survival for up to 30 days. So this was something we later introduced in our living building material. So to try to understand a little bit more about how these cells are surviving after desiccated, we also tried different types of parameters that would be important for a photosynthetic organism survival, such as temperature and light during both storage and recover. And if we see here in this video, in our initial conditions where we didn't optimize the temperature or the light of the recover, you can see that the cells here completely lose their chlorophyll fluorescence, showing that they are inactive and possibly dead. But once we optimize the conditions for recovery and storage, we were able to highly increase the survival of the cells. As you can see here, they are happily dividing under these optimized conditions, even after being completely dried after 7 days. So next, we're gonna talk a little bit about these biomaterials that we're interested by from being produced by our bacteria, and I'll switch to our civil and environmental engineering, Rollin. You're mute.
You're muted.
Hi, so I am Rollin Jones. I'm a first-year PhD student working on this living building material project. Today I'll be talking a bit about how we integrate microbes into materials. We've been investigating new strains and working to form biopolymers in addition to the biomaterials discussed earlier. So first, I'll give a quick recap of our living building materials and highlight an area of improvement. Our living building material is composed of cyanobacteria, the calcite minerals they produce, sand, and gelatin as a hydrogel. So gelatin does its job well. It helps bind our aggregate together, contributing strength to our material. Additionally, it provides a nice environment for our cells to live in and greatly improves their viability. However, gelatin is not favorable as it is an animal product, and I think we'd also much prefer autonomous gelation via the microbes over addition of exogenous gelatin. And so we're looking at other cyanobacteria for both mineral production and autonomous gelation. So 𝗕𝗂𝗶𝗴𝗆𝗆𝗆 has done great things for us, but those great things have been limited to producing biominerals. We have been looking at this Anabina strain as it has a few more interesting things going for it. In addition to producing calcite, Anabina 33047 can produce high concentrations of EPS, or extracellular polysaccharides, or EPS, and we hope to replace gelatin with EPS. Anabina 33047 has some other things going for it. Sorry, Juliana, can you go back one slide? So yeah, Anabina 33047 has some other things going for it. It is fast growing and can fix atmospheric nitrogen for itself. It is also filamentous, which may have some implications for viability and binding in living building materials. Next slide. So again, we're trying to replace gelatin with this autonomous biopolymer. The idea is to use EPS. Here we are taking inspiration from biofilms, of which extracellular polysaccharides are a major component. and provide cells adhesion, structure, and protection in our living building materials. This Anabina strain with high EPS production is being investigated for its ability to bind aggregates. This image on the right shows a very globular culture of Anabina 𝗕𝗏𝘴𝗎𝗫. Okay, next slide. Here's a quick video of our Anabina culture. We left this to dry on the benchtop for a week or so. You can see it is very gooey and globular. 𝗕𝗏cie𝗴𝗿𝗺𝗀, we've nearly created flubber. Now I just need to engineer some sentience in it. So if anyone could figure that out, that would be pretty cool. So yeah, anyway, I mixed this flubber with some sand and we let it dry for a bit, and this resulted in a nice little EPS brick. Next slide. So I took the EPS brick over to the SEM and got this nice image on the right. So yeah, on the right, this is an image of our EPS brick, and I'm comparing this to an image of a brick bound with gelatin on the left. We can see the cells in the EPS bridge sand particles in a similar way as gelatin. It was very cool to see the filamentous cells adhered to the sand surfaces and embedded in our EPS bridges. This also makes us kind of wonder about how the cells may contribute to the mechanical properties. Next slide. So then I used the EDS just to reinforce the idea that we were looking at EPS. On the right are EDS maps for silicon and carbon. The blue represents the silicon, and then the red represents organic carbon. So we confirmed that we're looking at an organic carbon structure. Next slide. So now that sort of proof of concept work is complete, we aim to start building Anabina 𝗕𝗏𝘴𝗎𝗫 into bricks for mechanical testing. We will be looking at different gelatin cell and EPS ratios for optimal mechanical performance. And additionally, we would like to further investigate interactions between EPS and calcite, and to see if EPS increases calcite formation. Okay, and with that, I'll hand it off to Bruce. He'll talk a bit more about material science, mechanics, and some of the cross-linking work he's been doing.
Hello, everyone. My name is Bruce. I'm a PhD in mechanical engineering, and my job in this project is to make various shapes of construction material with a living building material, and also the corresponding mechanical tests. And Joanna, can you move to the next slide? Yeah, if you look at the left picture, this is the way that how we made this living building material brick. We use a plastic mold, as you can see on the top, to put the sand and the mixture of the gelatin and the seal and the calcites together. And after the cure, we just demold it and leave the brick, as you can see, the cubes, in the room temperature for about 8 days to let it dry until there's no mass lost. And also with a similar technique, we can make larger bricks, as you can see in the middle, and also complex shapes, as you can see on the right side. So the material that we make, as you can see on the bottom pictures, has a good processability, that we can use a diamond saw to cut a nice notch, as you can see on the very red bottom picture, and the bricks or the particles will not fall off. And this is one of the sample that we used for mechanical test. We also follow the ASTM C109 for all the mechanical tests, and theoretically, since it's a material that we can build up, we can use a 3D printer in the future to build various shapes. So for the next, yeah. And so here I'd like to talk about a recent work that we did for one of them is the cross-linking of the gelatin, and the purpose of doing this is we are trying to offer a better mechanical property such as stiffness and water resistance. And another one is we are looking for some alternative biopolymers instead of gelatin. As Rollin just mentioned, gelatin may not be the best product since it's an animal-based product. Well, for the cross-link of gelatin, we use safe, or actually a food-grade enzyme-based cross-linker, which is called 𝗕𝗶ecie𝗴𝗿𝗺. It is completely food-grade, and you can find this in the chicken nuts or something. And the mechanism of this cross-linking is quite straightforward. It's just cross-link some residues in the gelatin and produce a little amount of ammonia, which has already been proved to be safe to our cyanobacteria. Well, another part of the alternative of biopolymers we are looking for is the polysaccharides. The reason why we are looking for the polysaccharides is, first of all, it's a good binding material, just like gelatin, and also it's a non-animal product, and as well as the good mechanical property that it can offer. The polysaccharides that we explored is first of all the 𝗕𝗶ecie gel, and the second is the carrageenan, and the third is 𝗕𝗏carrageenan, and these are all widely used in cultivating the bugs and the cells. And we also proved that they have a good compatibility with cyanobacteria 7002. So in this research, we actually not completely replace the gelatin with polysaccharides. We actually only replace 20% of that. The reason why we're doing this is that gelatin still pertains a vital gelization property. Remember that I just talked about that we have to put it into the mold, and after to cure before we can demold. So with gelatin in the system, it's better for the for making the breaks. Well, if you move to the next page. Okay, so we carried out a mechanical test of the break made with living building material. As you can see here, there's a break here sandwiched in between two mantle plates. The break is made of living building material, and the two plates are from a loading machine, and they kind of compress the break until it fails. At the same time, the load will be recorded, and we can get the maximum loading. Well, in one word, the higher of the maximum load, the better the mechanical property is. So then we get get this data, as you can see on the right side of this page. So first of all, we have four different colors representing four different categories in terms of the material that we use. So the green represent that we use gelatin as the only binding material, while the red represent that we use a combination of the gelatin and the carrageenan as a binding material, while the blue representing the gelatin and 𝗕𝗏ecie gel, while the pink representing gelatin and 𝗕𝗏carrageenan. Well, and for each category, if you look at the x-axis, we have four groups. We have the two control group on the left and the two cell group on the right. In the control group, we only have the biomaterial, which means we don't have the cell, we don't have the calcites. While on the cell group, we have everything, including the bugs and also the calcites. So for each group, either the country group and the cell group, we have a subgroup of the cross-linked one and the non-cross-linked one. Since in all these groups, they have gelatin inside. So if you look at compare the column between different colors, for example, the first four columns, you can see that actually adding a little bit of polysaccharides into the system will slightly reduce the mechanical property. However, it's not a dramatic reduction. Well, if you look, compare the same color, which is the same category of material, but in different group, it's very obvious that the cross-linker has a very obvious enhancement of the mechanical property as well as the cell. While the mechanism of the cross-linking is to make the polymer network more dense, thus have stiffer mechanical property. However, for the cell, since the cell produce tons of calcites, and those macro scale calcites in a macro scale way will enhance the binding material better. So there are different mechanisms, but they all offers good or positive improvement of the mechanical property. Well, the polysaccharides in one word offers a slightly kind of reduction of the mechanical performance. However, consider it's a non-animal product. I think it can also be a good alternative in the future for our living building material. And apart from this mechanical test, if we move to the next page, we also carry out some other tests, which is an in-water test. So the aim of this test is we want to explore how this breaks behavior in extreme conditions. So in this experiment, we just simply put all the breaks in the water. We immerse them in the water for about 1 week. As you can see on the left picture, the top 3 breaks are made with gelatin that is not cross-linked, while the bottom 3 are made with the gelatin that is cross-linked. Those without cross-linked gelatin at about 1 week has already shown deterioration, or the shape has been fallen apart, while the cross-linked one has already still maintains this cubic shape. And at the same time, we also keep a track of the mass gain. As you can see on the right side of the figure, we keep a track of the mass that they gain with respect to the time. So as you can see on the black curve represents the one with the gelatin with non-cross-linker, and while the red curve is the cross-linked gelatin. So we observed that even if we cross-link the gelatin, it's still absorbing a lot of mass from the water, and this is because cross-linking the gelatin only stabilize the polymer network, but it doesn't necessarily changes the hydrophilicity, or in other words, it won't flavor from hydrophilic to hydrophobic. So keep this in mind. We carry out another test. If you move to the next page, which is a wet-dry cycle test. So the test is really similar. We still put the breaks in the water for a certain amount of time. Here, as you can see, we put the breaks of both the cross-linked one and the non-cross-linked one in the water for 1 day, 3 days, and 5 days respectively. And we pull them out and dry them in the air, the room temperature for almost the same amount of the time. And after dry, we use the same machine, which is we carry out the compressive mechanical test, and we compare the mechanical property with respect to compared to the original one, which have never been immersed into the water. So if you look at the left picture, which is the breaks with the cross-linked gelatin, after 1 day immersing in water and complete dry, it still maintains about 25% of the mechanical property, while for the 3 days, there's still like 11%, and 5 days, about 6 to 7%. And compared to the cross-linked one, if you look at the right side, which is the non-cross-linked one, after 1 day, it only retains less than 60% of mechanical stress, not to mention the 3rd day and the 5 days. So this test is to prove that the cross-linker in the system will enhance its stability so that it will improve this mechanical property instead of changing the hydrophility or hydrophility of the brick. And if we move to the next, there's some up-to-date things that we are doing. Right now, we are trying to mixing the fibers in the building material. This is our most recent updates, and I'm still working on this and to see what kind of potential improvement it will have to our living building. And okay, that's all my updates, and I will move hand over to Juliana again. Thank you.
So as we were both talking, we were all talking, you can see here in this SEM of the LBM being produced, where we can see that the area between the gel and the sand has clearly precipitated calcium carbonates. And we know that the calcium carbonates can bind to the sand, but I just wanted to make sure to show you that this is clearly being seen. And as you saw, we had an improvement on compression, but we also have an improvement when we look at this 3-point bending experiments. They're done by the engineers, where you put pressure in the middle and you see if these bricks will break. This is important to measure impact resistance of the material. And if we compare the samples with simply the chemical precipitation of calcium carbonates by simply raising the pH or just in the media, we can definitely show that the bacteria in Micp have an increased effect on the fracture energy, as we showed before. So later on, we wanted to try to tailor more these LBMs by manufacturing the conditions, by changing amounts of gelatin being added, or changing the bacteria and the metabolic process being used. We talked a little bit about using different biopolymers, which we still are working and studying, and introducing different fibers on these materials. But simply by adding different bacteria through different biological process, we saw that we could get a very increased amount of change in our flexural strength, again, which is related to this higher impact resistance with the Micp, both by photosynthetic or urolytic pathways in this bacteria. And these flexural strength were even comparable to high fiber bricks or even cement mortars, which is used to compare for the engineering purpose. We also tailored with concentration of gelatin by adding more or less bacteria, gelatin, and Micp, and trying to tailor our LBMs for whatever the consumer later will be using in their construction material. And we saw that by adding our trehalose that we talked about, we were able to highly increase the survival of the bacteria on these bricks that were dried for such a long time and kept under ambient conditions with low relative humidity and high temperature. So this seems very attractive for us in following up with new different ways of trying to manufacturing these LBMs, both by using biopolymers and integrating later with these fibrous materials that Bruce was talking about. So a little bit conclusion. So I hope we showed to you that we can use these organisms, microbacteria that I know it's widely using in this seminar, but we can use them by producing biomineralization and also biopolymers, which we can make this using the hydrogel to make these nice living building materials, with which we can tailor how we manufacture them to increase the cell's viability on it, and structural integrity by increasing the mechanical performance. This material is also easy to construct. It's malleable. We can use different shapes, different structures. It can be cut, reassembled. And we didn't talk much about here, but we showed in our previous paper that by using the same brick, we are able to regenerate the cells using the same material just by adding a little bit more of media for the cells to grow and calcium carbonates and calcium to produce calcium carbonates. We were able to make these bricks 3 different generations following up. So we're still working in our next steps are increasing this durability and the service life. So we like that our material can survive in different habitats and where we can reuse the same cells. So that would be avoid some waste and recycle of the material. It would allow some recycle of the material. And our next steps are also involved in scaling up this process so we can actually use this in construction sites. So right now we are increasing the size of the bricks, trying to find ways of keeping the bacteria alive, which can be sent to anywhere in the world or extra world. And with that, I want to thank you. I hope we gave you a little bit of a glimpse on our multidisciplinary team. It involves different engineering departments from civil, environmental, and structure, and together with the biological professors. We had several former members that we want to thank for their results here in collaborations, and we had funding from DARPA. With that, I thank you all for your attention and we'll take any questions.
Thank you very much. That was really awesome. I think you have a lot of questions in chat. I think Bruce was trying to reply to some as you go, but if you want to just look at chat and go through the questions.
Okay, so the first question was, if the material is made from cell matrix, will it not be digested by bugs if it's used as a building material? That's a great question. We actually have been looking into some other bacteria that could, be living later on our bricks, because it has a high gelatin, which is rich in carbons. And we haven't done too much tests yet in field, which is something we want to do, but from our little experiments, we have a little bit of bacteria being contaminating, and that's one of the reasons we're looking at different biopolymers. The cell itself, at least the cyanobacteria, it wouldn't be consuming the gelatin, so that's one of also advantages we look into that bacteria. The second question would, what is the tensile strength of the EPS bricks and other type of materials you make? So we haven't yet measured tensile strength for EPS bricks. Those are underway, so hopefully in the future we'll be able to answer that question better. Another question is, can you engineering cells to make materials? What properties of synthetic cells would you like to see in an ideal building material? Well, that's a good question. Well, we would like to see the bacteria producing its own hydrogel. There are some efforts, that different people use different material. Our bacteria, the cyanobacteria that Rollin mentioned, Anabina 𝗕𝗶ecie𝗴𝗿, has an advantage of producing a lot of EPS, and EPS have been shown to also be related to calcium carbonate production. So we're trying to make the bacteria by itself being able to produce these assembling kits. Of course, that's our goal in the future, but we're not there yet. And we can synthetic engineer the cells to produce stuff like different types of EPS or halos or whatever. but we would like to have a more natural pathway by investigating different strains also. But that's a possibility that we will definitely explore. We're exploring. Another question was, what are the advantages of cyanobacteria Brix over mycotecture? Sorry, I'm not. I guess they're talking about using fungus. Mycotecture is making brick from cyanobacteria better than using fungi intrinsic structure for hardness. So there are some companies that are investing in fungi, and I personally saw some of the bricks. They're pretty interesting. You can step on the top of them, and they seem really reliable. And I think you know, the advantage of cyanobacteria, each of them have their own advantages. Cyanobacteria we like because they consume, you know, CO2 from the environment. There are some suggestions that we know that when precipitate calcium carbonate, there's formation of CO2, but the cells would also be able to consume that CO2. So we would have a more sustainable type of environment, and that's why we specifically were looking to cyanobacteria. Next question is, as polysaccharides tend to absorb water to a certain extent, considering varied environment conditions, rains, won't the bricks lose its strength or disintegrate into components? Thanks for that question, and I hope that some of Bruce's experiments answered some of those questions. So, you know, we're not thinking of our bricks definitely being underwater. That's not exactly what was our initial thoughts, but I hope that his in-water experiments show that we can tailor the structure of the bricks by, for example, adding cross-linking in the hydrogel to increase this capacity to survive during rain. And another question about sand, basalt, etc. mixing. What's the advantage of mixing it with cyanobacteria instead of just building with sand, basalt, and etc.? Sorry, I don't know if I understood your question very well, but I think you were talking about using different types of sand. If that's what it is, our objective would be using whatever sand is in the local, so with minimal treatment. And we're still needing to explore other types of materials, like, but that will be our future idea. Can you use multicellular organisms for construction material? So that's something that Rollin talked about a little bit. So I hope we are exploring that opportunity. So that the idea would be that, you know, once, since we have multiple cells already there, they have their own strengths. So they have different cell structures. They can produce different concentrations of EPS. Some of them can produce trehalose and other biomaterials that are of interest for us. So that's why we're exploring also this other material. And in the case of Anabina 33047, it can actually grow even faster than our model organism, the 702. Why do we care about cell viability? Would it not be possible to grow the bacteria first and then shape it when the cells are dead? So yes, there are some studies that actually show that dead cells, by being present in the bricks, they can also increase concentration of calcium carbonate. I think this area is still under study, and you know, we'll hopefully know more about it in the future. But in our case, we were kind of interested in having cell viability, so because we call it a living building material, which would be able to recycle without having to, you know, obtain the material source again. But again, like I mentioned, we can tailor this our bricks depending with what in the end the customer will be using for their own construction. So if they're not interested in self-viability, they might not care about adding, trehalose, for example. And could you use cyanobacteria mixed with other soils, for example, 𝗕𝗂𝗶𝗃𝗃 for construction of habitats on Mars? Yeah, that's an interesting question. I think we would have to be able to test it. Construction on places outside of Earth or of extreme habitats, like as we mentioned, it's definitely something that we are interested into. We think that yes, we possibly would be able to, especially because we have very good results with our 3D printing by using droplets. Our material is very easy to mix with whatever we want. I can't give you concrete results on their mechanical performance, but like I said, we were probably able to tailor the bricks for whatever construction needs. That's our hope. And last question, how do those materials weather in snow and freeze-thaw cycles? That's a good question. We haven't looked too much into high cold temperatures. We looked more into keeping the fridge at 4°C. And actually the cells are very robust, both the E. coli and the cyanobacteria. They can survive pretty well. And as I mentioned, they can recycle pretty well. In freeze-thaw cycles, we would still need to be checked, like in very extreme low temperatures. But, like if you're not interested in cell survival, or we would need to be able to test this, but we haven't looked into it. Oh, and thank you for someone sending a possible collaboration. Yeah, I will definitely send that to our PIs. And if there's no more questions, thank you so much for listening to us. I hope that 3 speakers wasn't too overwhelming, but give you, a nice idea of how multidisciplinary this project is and how important, it is by exploring science in a way that we can make more sustainable materials for a sustainable future. So thank you very much.
Thank you all, and thanks everyone for the great discussion, and thank you for presenting. And see you all next week.
Thank
Bye,
you.
everyone.
Thank you. Bye.